For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started. All right, guys, looks like we are live. And I just realized, actually, I forgot to change my name there. It's currently Professor Dave versus LPP, but I'll change that in a minute. I'm excited for this. This is going to be a good night, guys. It's nice to see that we got a good chat already. Now, I know a lot of you were excited for tonight's debate. It was supposed to be myself versus David Beldock on universal common ancestry or independent origins. Actually, I'm going to check the chat real quick, guys, if you want to give me an audio and video check. I was having slight issues with my internet over the last couple hours, so it should be good, but just in case there are any issues, let me know, and I'll do what I can to save some bandwidth. So looks good right now, but keep me posted. So the debate between David and myself, thank you, Patrick, it's been rescheduled for next Saturday, same time, 9:30 EST. So same topic. David Beldock came down with a flu. Very sweet of you, our good friend Speed. So good to have you in the chat tonight. Speed, of course, when we open the mic, you are welcome. And everybody's welcome for the open mic. We're going to be starting with a couple discussions. It's supposed to be myself and a rational empiricist here. We're going to talk about evolution, common descent, universal ancestry, independent origins, all of those related topics. And then we're supposed to be having David Neff supposed to join us later for a discussion on human evolution, out of Africa theory. versus the out of Middle East theory. So once we're done that, though, probably a little bit over an hour, then we're going to open it up for an open mic tonight. Should be fun. Tomorrow, guys, we have Professor McQueen back with us. It'll be George, myself, and Professor McQueen. It's going to be around 6 p.m. EST. So that's going to be a lot of fun. Make sure you are here for that. And a ton of good debates and discussions and interviews. Too many to go over here. I don't want to bore everybody, but go check the upcoming live stream section. I think we've got about 7 or 8. A lot of great debates for the end of the month. Professor McQueen versus Jordan from Reasons to Doubt podcast on accelerated nuclear decay. T-Jump versus Bill Morgan next week. We're just finalizing a date and time for Dr. Ron Garrett's going to be back with us discussing the Ark. Is Noah's Ark real, or is there evidence for Noah's Ark with Nephilim free? So that should be a lot of fun. This Thursday, we've got Fighting Back. He'll be here to debate Boy. Boy's been here a couple times. Actually, Boy will be back on the 2nd of April as well to debate Nephilim free. So that's just a few exciting debates to look forward to. Like I said, go check the upcoming live stream section. I'm trying to keep that as organized as possible for everybody because we've got a ton of shows, and that's probably the best way for you guys to remember all the different days and times for everything. So anyways, enough for me. Rational Empiricist, him and I, we've had a few discussions in the past. I always appreciate having a cordial, respectful discussion with Rational Empiricist. So this is going to be fun, and I think a good way to start off the night. So Empiricist, thanks for being here.
Yep, pleasure to be here. Can you hear me? All right. Is my audio coming on okay?
You sound good.
Oh.
So we have had a lot of new subscribers lately. So, I mean, why don't you give yourself just a brief introduction as to who you are. We have had a debate over a month ago now. I think time seems to fly by.
Yeah.
Especially these days. So most people are familiar with you, but if you want to give just a really brief introduction into who you are you thinking about making a channel in the future, since I know you're more active on YouTube now? So a brief introduction. What's going on?
Yeah, my name's A Rational Empiricist, or I like to call myself that, and I just like to discuss science and evolution on the internet with basically anyone I can talk to, because I love discussing these things. I don't know if I'm really gonna come up with my own channel. I don't know if I have the mechanics of that figured out yet, let alone the tech part of it. So but that's pretty much it.
Well, if you do start to if you do decide to have a channel one of these days, you'll have a ton of content to just throw right on there, because I know you've had a lot of discussions and a few debates already, actually. So why don't we get right into it then? We're going to start off with, as I was explaining earlier, just kind of evolution, hierarchies, similarities, common descent, all that fun stuff. And we're going to start off with Rational Empiricist, who is the guest tonight. So we're going to allow him to just take as much time as he needs. He's going to go over, I think, a few slides, a few of his arguments, and then we're just going to have kind of a back and forth on the topic until we've exhausted the topic that we're talking about tonight, and we'll kind of go with the flow. So Rational Empiricist, good time to start. We got 30 people in the chat, so enough with introductions. Let's get right into it. If you need to share screen or anything, let me know. And there we go. It just popped up for me. So
let's see. Is it? Does it look all right? Let's see. It's loading. Okay. How's it look? I can't tell from here.
To me, it's just a black screen right now.
Oh yeah. All right.
But there is something, so it looks like it's working at least.
Okay. So the topic I want to discuss tonight is, how do animal bodies evolve over time? And the answer, I think at least, is changes in development. And this is specifically the field of evolutionary developmental biology, also called evo-devo, for all you cool kids out there. So first of all, why is development important? Animal bodies, first of all, grow and morph in a sense from an embryo to an adult, form over the course of their lives using key developmental pathways that are genetic, of course. These pathways are controlled by regulatory genes. You may see different terms for those kinds of genes, but I'm just going to call them regulatory genes here. They themselves can change, or the switches that promote / inhibit their expression can change. So that's how new physiology can emerge. And for instance, here is a diagram of the segments of the hypothetical common ancestor of arthropods. And you can see that some of these segments are changing over time. And that's thanks to developmental genes, regulatory genes. And you can see how they're different in this velvet worm, this probably a millipede or centipede, this wingless insect, and of course this Drosophila fruit fly. There are specific types of regulatory changes that I like to go on. First, there's changes in the location of expression called heterotopy. There's also changes in the timing and rate of expression. That's called heterochrony, chrony for time. And finally, the amount of expression, that's called heterometry, metry amount. So changes in the location, timing, and amount of expression of particular developmental genes can create very obvious changes in the growth and morphology of organisms, and they can be quite immediate. So here, for example, is a picture of a, we can imagine this is a developmental gene, and this is RNA polymerase binding to a promoter and transcribing those genes. And also we can see these homologous structures between the bat and the mouse and how they are simply modified versions of the same template, basically. So first, an example or examples of heterochrony, changes in timing. So for example, the nose, changes in the nose and jaw genes of dog breeds, of different dog breeds, lead to different dog faces. So you can see well, I guess I'll pull up that image in a sec. There's also changes in the timing of the genes involved in the toes of horses. So enhanced growth of the central digit and reduced growth of the lateral digits leads to a single hoof like we see in modern horses. So here is that dog thing right here. You can see they're due to different differences in timing and rate of the growth of the dog faces. So we have Cocker Spaniels here, we have a Bulldog, we have Wolfhound. I don't even know what dog breed that is. So we also have examples of heterotopy. So for example, you have changes in the expression pattern of pigment genes that lead to different coloring patterns on different animals. You also have changes in the location of certain bone genes. So this can lead to, for example, the panda's false thumb, and also certain bones, or let's see, why can I not remember the name? Certain bones in the tails of theropod dinosaurs and probably some other kinds of dinosaurs that help with balance. And finally, ducks likely got their webbed feet through this protein called gremlin being expressed in the hind feet. So gremlin inhibits apoptosis, programmed cell death of the webbing. And so when it's now expressed in the hind feet, the webbing grows between the toes. So now the hind feet are webbed of the duck. So here we have human hand and the false thumb of the panda. And here we have the gremlin changes I was talking about. And finally, we have examples of heterometry, or rather, one example, we have changes in the timing and the amount of expression of BMP4 protein in various species of Galapagos finches. And this has been shown to result in their different beak shapes. So, for example, ground finches have deep, broad beaks for cracking seeds and nuts, and cactus finches have long, narrower beaks for getting into cactus flowers and getting pollen and nectar and insects, for instance. So, in conclusion, how do animal bodies evolve over time? Animal bodies evolve through changes in the spatio-temporal expression of developmental regulatory genes. So that's one way. And there are many examples of these developmental changes, as I've shown. Differences in the genes and their expression pattern lead to differences in morphology, and many are still being studied, of course. And so overall, proteins aren't always necessary for morphological structures to change. In fact, it's more so the regulation of these developmental genes is what's actually involved. So thank you for tuning into this presentation, and I guess I'll see you whenever I do a next presentation. All right, so I'll stop presenting there.
Awesome. Andrew, you can hear me?
Yep, I can hear you.
Okay. All right, perfect.
Uh-oh, I got an echo.
Oh, let me see real quick. How's that sound?
Let's see. Okay, now it's better. Huh.
Okay. No problem, no problem. I had my volume all the way down because instead of muting, as we were talking prior to going live, we have that issue where there's that roboting if I mute myself, but with my external mic, I'll just decrease the volume while you're talking. And as I'm increasing it, there might be a slight echo, but yeah. So I'll say a few things. A couple things pertaining to your presentation that come to mind, but also a couple things just in regards to developmental windows and shared patterns in these different developmental stages, as well as when it comes to evo-devo, when it comes to the control, the very precise control of these genes, okay, that are involved in development, okay, because you pointed that out in your presentation here, and I appreciate the visuals. For one, I would say that the descent part, okay, in terms of the conclusion of what we're seeing in developmental genetics, is it consists of countless assumptions, many of them not even justified by the empirical data that we have today, which we can talk about. But I would say, given the control and complexity that we see in developmental genetics, it's difficult, and I would even assert impossible for the evolutionist, the naturalist, okay, in terms of these new novel layers of complexity and information and control, okay, raise more problems, raise more questions rather than more answers. Now, I do want to point out, though, that similarity, because oftentimes, like in our previous discussion or debate, Andrew, we talked about homology, transitional forms, nested hierarchical patterns, for example, and how they are agnostic to the debate, because both models can explain the data and both models expect the data, right? Both models expect homology, nested patterns, as well as transitional like forms or mosaic creatures, I should say. But we also see shared patterns and similarities on the embryological and developmental stages. Okay, this is a fact as well. This is something we can observe. We actually see shared patterns in the early developmental stages of various forms of life. Okay, we could look at a large diversity of creatures. Okay, let's say turtles. We could look at chickens and say fish. Okay, and what we find are very similar patterns and very similar features during these developmental stages, these developmental windows. Okay, primarily in the early stages. Okay, the later stages though are extremely different, okay, than the earlier stages. That's where we see the greatest differences, is at the later stages of development. And as I've pointed out with homology, nested patterns, I'll show a couple visuals in a second to give some of the viewers in the audience that may not have seen our first exchange. I'll show them what I mean by how we, the evolutionists and the creationists, would both expect, okay, these lines of evidence. But we also see the same thing in the design world when it comes to these developmental stages and the similarities and the shared patterns. Okay, so for example, if you were to visit, let's say, a diversity of vehicle factories, okay, from say Toyota to Chevy to Volkswagen or Mercedes, okay, you would find numerous similarities in the early stages of manufacturing, and you'd find shared patterns even in blueprints. But the thing is, it's not until the final products that we find the largest differences. So there's an echo, once again, in the design world, okay? That echo is shared or observed in the biological world. And as I share a couple slides here, just one second, I'm going to share a screen. And how's the audio coming in? It's coming in okay?
Yeah.
Okay, just one sec. Okay. So I'm going to share screen here real quick. I'll say a couple more things and then we'll kind of just open it up and take it point by point. So here we go. Okay. So when it comes to, here we go. So when it comes to the homology or the transitional like forms, hierarchies, not only in terms of anatomy, not only in terms of morphology, but we can see hierarchies on a genetic level. We can see mitochondrial DNA hierarchies, nuclear DNA hierarchies. We can see hierarchies when it comes to these amino acid trees, okay? So the hierarchies exist. Homology exists, okay? Just through comparative anatomy, homologous structures abound in the biological world, okay? And if we were to look to some of the mosaics, okay, and we can get into this too, you could look to Tiktaalik, or you can look to Archaeopteryx here. You can look to the synapsids, okay, the mammal-like reptiles, for example. And you see this wide diversity. Now, when it comes to creation model, since we are made in the image of God, we should then hypothetically be able to get a sense for the way God may have designed the biological world based on the way we design things, based on the way we build things, okay? Because the Bible says we're made in the image of God. So that suggests that we reflect something about the design, okay? Now, obviously, theologians have debated what exactly does it mean to be made in the image of God. But one thing we can all at least agree on is that there is a reflection of us, a reflection of the divine, a reflection of the creator in us. And it just so turns out that what we see in the biological world, we see an echo of that in the design world, okay? Humans build in homologous patterns. This is good design. Humans build transitional-like forms. I mean, you've got an amphibious assault vehicle, which essentially blends the traits or blends the features of a military vehicle built for the ocean and a military vehicle built for the land. Okay, this is good design. This is good design. With the invention of your crossover vehicles, okay, you've got like the Dodge Journey, the Ford Flex, the new Jeep Gladiator. You know, these essentially resist class classification. And what's funny is God knew this. He designed the biological world, okay, like this in the beginning. And we are as human beings, in our finite minds and finite understanding, we are just in the infancy of recognizing design. We are now just getting to the point where we, okay, as human designers, are realizing that these types of transitional like vehicles are good design. Okay, they serve a purpose. And the same thing in terms of nested hierarchical patterns. We see the same thing in the design world. And when it comes to development, okay, and these various developmental stages, the similarities in the early stages of development across a wide diversity of animals and creatures, we see a reflection of that in the design world. So the point is, these are all agnostic lines of evidence, okay, when it comes to creation versus evolution, universal ancestry, or independent origins. And that's why we need to look to those lines of evidence, okay, that can really only be true if independent origins is true, or if universal common ancestry is true. And that's where we can look empirically to, let's say, natural selection. We can look to mutations. Genetics is what's inherited in sperm and eggs. That's the best way to determine ancestry, right? Bones and fossils, they're not inherited sperm and egg, okay? They don't really come with genetics most of the time. And we can get into why fossils are a problem, because similarity in form doesn't always demonstrate relationship due to the existence of incomplete lineage sorting, convergent evolution, loss of traits evolution. We can get into all that. But if what we see, this is the last thing I'll say, if what we see today in terms of mutations and selection, right, evolutionary theory would say that natural selection acts upon random variation. The random variation is the result of mutations over time, genetic mistakes over time. Add diversity, right? A mutation essentially adds diversity because it's adding something that was not previously there. Natural selection can act upon that diversity and random variation. But the thing is, those mutations that are supposed to drive a fish to fisherman, they are deleterious. They're damaging. They're not taking things forward. Okay. They are essentially the destroyer and not the creator. So these historical reconstructions and assumptions of universal common descent remain just that, historical reconstructions that have no real validity, because what we see in the present in terms of the driving forces of evolutionary theory, okay, they cannot account for that large scale change from single-celled like ancestor to whale to humans. So there's just a lot of assumptions. And, we can talk about a number of different lines of evidence. For example, I would like to ask you about like taxonomically restricted genes, which is exactly what we'd expect if the independent origins model is what best explains reality. But I'm going to stop it there because we've touched on a number of things. And I'm going to hand it over to you. Let me stop my screen share. I'm going to hand it over to you, Andrew, for some thoughts, and we'll kind of just take it from here. It'll be more free flowing. So go ahead. What are your thoughts, Andrew?
So I guess responding to your presentation, I had 3 main points. Does my audio sound okay so far, by the way?
Sounds good. Sounds good.
Okay, just making sure. All right. So first of all, there are examples. You mentioned development and the analogy to cars, basically, but cars are kind of manufactured in a set efficient way, I would argue. Whereas, for instance, you see in whales, they develop and or baleen whales, for instance, they develop and they start having teeth, and then the teeth go back into the gum socket, and then they get the baleen, basically. Same thing with snakes, for instance, and same thing with these other vestiges in development, basically. And you don't see the same thing in cars or designed objects, basically. So that would be my first point. My second point would be I would agree that nested hierarchies can be explained by creation and evolution, or sorry, creation and common descent. Both are possible, but both are not as likely. They're not equally likely. So, for instance, we have a mechanism, for instance, of how common descent generates a nested hierarchy. So we know how genetics works. We know how genes are inherited, and populations inherit genes from one generation to the next. We don't know that same thing with a supernatural or a common designer, I should say. So that would be my second point, and my final point would be that you see these um, you talked about the mutations and how they're deleterious. Well, like for instance, we can see differences in the genes, developmental genes of, for instance, the Galapagos finches, and obviously the differences in developmental timing are and amount are helping them survive thanks to the different beak shapes they have. So I don't quite understand how that could be called deleterious. So those are my 3 points, and you can go from there.
Okay. So good points, and so many different routes we can take by everything you said. So what I'll do is I'll touch on a couple things, and then I'll kind of just direct it to something that maybe we can kind of discuss specifically. In terms of maybe the bigger picture of this, what's true, the design hypothesis, the independent origins model, or the model of universal common ancestry, descent with modification. So we would say from our starting point then, from the design perspective, the fact that we're created, we're made in the image of God, we would say that everything for the most part, but I also want to make clear that what we are seeing in terms of the fall and the result of the fall is death, extinction, degeneration. So, we would still expect a lot of broken genes. We would expect a lot of damage, essentially. So what was once functional could certainly have gone to a state of non-function due to mutation, due to degeneration. But overall, we would say what we're looking at essentially is purpose, is function, right? These apparent vestigial organs or vestiges that we see in these developmental stages, let's say when it comes to the whales, like you were talking about, we would expect to be a part of the overall design, the overall bigger picture, and also to be a part of those developmental stages leading to the end product, because they have essentially, the evolutionary community has taken the vestigial organ argument, where hundreds of years ago, I think they had a list of between 150 to 200 so-called non-functional vestigial organs. Now you could maybe argue one or two. Okay, they've actually redefined what a vestigial organ is, and I've got some quotes and sources for that to show it if we got to go down that route. But what we now see in terms of these vestigial organs is function, is purpose. Okay, now they've redefined it and said, well, it's lost its original function, or maybe it's co-opted a new function. But this whole co-option's never been observed, especially observed in terms of like your functional pseudogenes or ERVs, ALUs. We can get into that later. That's a rabbit trail. But we would expect function and purpose. They've oftentimes looked at the genetics of, let's say, whales, and they've assumed that what they were looking at, these specific genetic markers, were actually vestigial genes. No purpose, no function. Okay, the leftovers of past common ancestors. But what they now know is that a lot of these so-called evolutionary leftovers or genomic fossils, because that's what the common descent model would suggest, that we are looking on a genomic level, we are looking at a lot of genomic fossils, evolutionary leftovers. Okay, that would be a prediction of evolutionary common descent. Well, we would say that what we're looking at here has a purpose, has a function. A lot of these pseudogenes, for example, they have turned out to be incredibly essential and functional in terms of healthy life processes in the cell, oftentimes specific to the different organism. My question to you, though, when it comes to the changes that we see, because we've got these historical reconstructions and we've got these assumptions, okay, of universal common ancestry. But what we now know is that for example, would you agree, Andrew, that the genome, okay, the set of instructions, okay, that make up the genomes of living organisms, would you agree that they are not just a simple linear array of letters? Would you agree with that misunderstanding years ago? One gene, one protein, one function. What we now know is that the genome set of instructions are dynamic, they're nested, they're self-regulating, they're multidimensional. Would you agree with that?
Yeah, I mean that's pretty obvious, so I would agree with that. Yeah.
And that's the problem, is when we're looking to mutations as the source for all genetic diversity, okay, to take your single-celled like ancestor into a whale, into a human, well, this type of goo to you evolution, this type of fish to fisherman evolution has to explain then, okay? It doesn't have to just explain this erroneous view that the genome, the set of instructions is just a linear, very simple array of letters. No, it has to explain how this four-dimensional genome, okay, with multiple overlapping codes that are nested, okay, and are chock full of meta information came about. And that's why these mutations that come about, let's say roughly in the nuclear DNA, 100 new mutations per person per generation, okay, in a genome that is four-dimensional and nested like this, okay, it is impossible to say really with any certainty that a mutation can be completely neutral, have no effect on genotype. No, the chances are these mutations that are changes, random changes in the nucleotide sequence, are going to have a damaging effect. Okay, they are going to break something rather than make something better. So that's what I mean about these historical reconstructions and assumptions, is what we see in real time. These mechanisms that you're purporting explains large-scale evolution. What we see today and what we know about the genome, it's very unlikely. Yeah, go ahead. What are your thoughts on that? What do you disagree with in terms of what I said? And we'll kind of go from there.
Okay, so I guess a question I might ask would be, you would agree that a mutation is defined as a difference in the nucleotides of the DNA or whatever genetic material you're talking about, but mostly DNA, right?
Right. Yeah. I mean, there's different types of mutations, right? Frame shift mutations, point mutations, duplications, of course. So like a point mutation can be just a change in nucleotide from one nucleotide to another. But essentially, yeah, mutation I would define as a change in the nucleotide sequence.
Right. So any, I guess my position would be any change, or I should say any difference in these, like specifically, I think we're talking about cis regulatory modules, basically these regions near the gene themselves, and when certain proteins bind to that region, the gene is either transcribed or it isn't transcribed, or it can also be various degrees of transcription, basically. So basically, if we see differences in these regions, we have a pretty good idea that there was a mutation there, because otherwise you can't have, for instance, sexual recombination can't, I guess, reshuffle the cards and get that, basically. But like, so any differences we see, we have to say that they're due to mutations ultimately at some level. So I think that I guess what are your thoughts on that to start off with?
Just a second. I'm just going to screen share for 1 second. So good points. There's a couple of built-in assumptions to what you said in terms of You're correct. Any genetic difference, any letter difference, okay? The evolutionist assumes that all DNA differences are the result of mutations over time, okay? Well, the creationist starting point is a starting point of design diversity, okay? Created nuclear heterozygosity, as I say. Where the majority of DNA differences, based on what we know of the function of the genetic code, based on what we know about the multi-dimensionality of the genetic code, we would say that the majority of DNA differences are actually the result of created nuclear heterozygosity. So they're there for a purpose. They're initially front-loaded by God at creation, which essentially demands nested hierarchical patterns in DNA, okay? But that's why we would look to the genetic code, and this is a direct prediction of that model, as being functional. Okay, with obviously some damaged genes. We've got genetic entropy at play. So we would say, you know, probably between 60 and 85% functional. But specifically, these genomic fossils, these evolutionary leftovers, these ALU sequences, the ERVs, all of these things that the evolutionists say are the result of descent with modification, okay? This is where we specifically want to look to test the models and say, okay, is this all really just a long process of descent with modification, or is this the result of initial design? And that's why I'm saying, my point is, these mutations that we see today, okay, these mutations that we see today demonstrate that those historical reconstructions, especially in terms of the fossil record, okay, and these assumptions based on these highly complex control mechanisms that we see in developmental genetics that you're pointing to, you know, I would say that presents more problems and more questions than actual answers and solutions. But we can test these models, okay, by seeing if what the evolutionist assumes is a result of evolutionary leftovers, fossils in our genetic code of past common ancestors, essentially past evolutionary events, are they really just that? Right? Evolutionary leftovers and genomic fossils. But what we see is function, we see purpose, we see irreducible complexity, for example, like these. I had some more papers that okay. So I got a few here. I'm just curious as to your thoughts of these. You know what? I don't want to move to a new topic just yet. So what we'll do is we'll just start with pseudogenes, for example, because I've got paper upon paper here. Okay, so here's just 1, 2, 3. Here's 5 papers. None of them coming from creationist journals, creationist papers, but they are pointing out that these pseudogenes are actually functional and essential for healthy life processes in the cell. I mean, you can go over all these papers showing that they have multiple diverse functions. Right here, it says that they can affect not only their parental genes, but also unrelated genes, right? It's this regulatory genomic network that we see in the genetic code. And remember, these are what they looked at, these non-functional pseudogenes, genetic scars, genomic fossils, to say that creationists can't explain. Why would God create genomes of living organisms with genetic mistakes that are shared across species, right? Specifically humans and chimpanzees, they'll look at these pseudogenes and say we inherited them from a past common ancestor. But based on what we know on how highly functional and essential they are, okay, and regulating to the genome, mutations then are going to break things rather than build them up. So how do you explain what evolutionists once assumed was the result of evolutionary leftovers? How do you explain the function of them? How do you explain the fact that they are essential to the genomes of living organisms? I mean, how did that come about? You know, do you have any empirical evidence or technical papers that can show like a pseudogene or an ERV or something like that, or even just a genetic sequence of the non-coding region popping up and becoming a taxonomically restricted or orphan gene? You know, these types of things. I know I said a lot there, so take your time. But the main question is, you know, how do you explain this empirically from the evolutionary model in order to see if the historical reconstructions and assumptions of common descent that you have, let's see the validity of those. What are your thoughts, and how do you best explain the functional components of these DNA elements?
So, like, specifically, you're talking about the function of the pseudogenes.
Yeah, let's start with just to kind of focus on just one thing, because we could talk about a number of things, but if we could just kind of hone in on one thing, just the pseudogenes, for example, that was never predicted by the evolutionists, that were used as evidence, like the vestigial organs argument, used as evidence for common descent. How do you explain that what we see is direct expectations, really, and fulfillments of predictions by the design model? Yeah, how do you explain the function of these DNA elements?
Let's see. As far as I'm aware, we do have pseudogenes that aren't functional in that sense, but I think functional at this point, for one, is kind of relative and kind of controversial in the field right now, I think, but it's not my area of expertise. If it's okay with you, I'd like to address something you said earlier and ask a specific question.
Yes, yeah. If I think the one last thing that I would say to that is um, the fact that, because I guess the one point you made is that we have pseudogenes that are not, we don't really know the function of them yet. Well, that's because, for one, we're accidentally finding functions in these DNA elements like pseudogenes, ERVs, but we do have biochemical evidence that over 80% roughly of these pseudogenes, okay, based on the areas that they tested, have at least biochemical evidence for function. They have evidence for activity. Right? A lot of times the evolutionists want to say that this activity is spurious, right? That's a comment that Dan would often use. But without any serious experimental data, such as in terms of genetic knockout tests, right, which creationists are making predictions. Knock out this genetic sequence, knock out this DNA position, or mutate this DNA position, can result in damage, disease, very specific predictions on function. So I would say that the trajectory favors genome-wide functionality, even in terms of these pseudogenes. And the best argument for that is the fact that I find evolutionists, even your best evolutionists, struggle in explaining how these DNA elements went from non-functional to functional. I mean, I've been asking for probably almost 2 years now for just a single paper that shows a non-functional ERV going from non-functional to functional, right? Because they claim co-option. And I haven't been provided one. And I think it was Speed of Sound the other day who said in a stream that, why would we expect to have a paper like that? That's not something we can really observe. That's something that would have happened in the past. And that's the whole point. It's assumptions based on the past. It is assumptions based on the starting point of universal common ancestry, and they build these historical reconstructions. But the question is, are those historical reconstructions valid and plausible, and do they fit reality? Because when we look at the empirical evidence in the present, especially in terms of genomics, it pretty well just destroys the historical reconstruction. So anyways, yeah, that would be what I'd want to say, at least to kind of end that topic. Of course, if you want to respond to anything I said there in terms of the pseudogenes, go ahead, and then I'll hand it over to you if you had a question or a route you wanted to take it. Go ahead, Andrew.
Yeah, so I guess my question would be, so earlier I mentioned, or I guess first I'll start with this question. So you mentioned a common designer creating humans in his image, and then a nested hierarchy flowing from there, if I understood you correctly. Could you, I guess, explain that a little better? Because I guess my specific question is, like, irrespective of how evolution may or may not happen, like natural selection or drift or what have you, irrespective of those mechanisms, you would still have common descent, for instance, because we see organisms reproducing in a particular way, namely that what happens in X generation is for the most part, only dependent, or well, mainly dependent on the previous generation from X, for instance, and that's, for instance, how you get the nested hierarchy pattern um, that we've talked about so far. So I guess my question is, um, why would we expect a nested hierarchy from a designer, for instance? Like, and yeah, I guess start from there.
So essentially, based on everything you said, it comes down to why should we expect these hierarchical patterns in living organisms from the design starting point?
Right. I guess specifically I'm talking about, because we know that a designer could explain that, but what I'm looking for is why should we expect that, and namely, how do we generate that kind of pattern from that prediction, basically, is my question.
Well, why wouldn't we expect nested hierarchical patterns in the biological world based on the design hypothesis? I mean, is it really any surprise that we share more in, let's say, humans? Is it really any surprise that we share more in terms of morphology, anatomy, physiology, and genetics with a chimpanzee than we do with a fish or a bacteria? Of course, even if you were to just stand back, even before genomic sequencing, Andrew, you could just stand back and visually look at a chimpanzee and a human, and you would assume, wow, we probably share a lot in terms of the underlying system when it comes to genetics, whatever, morphology, anatomy. It would be easy to conclude just standing back and doing an assessment that, hey, we're going to share more with the chimp than we would with a dog. And just looking at the basic anatomy and morphology, then it wasn't really a surprise that when we we're able to sequence the genomes of living organisms, it just so turns out that we also share more on a molecular level with a chimpanzee than we do with a fish or a bacteria. That's why I say that, these lines of evidence from homology and nested hierarchies, they're agnostic to the debate, because essentially if the created nuclear heterozygosity hypothesis is true, okay, and that's the way God created. He front-loaded the original kinds. He front-loaded Adam and Eve with these pre-existing functional DNA differences, and front-loaded the first kinds with variation-inducing genetic elements. Okay, that's what we would call your ALU sequences, ERVs, pseudogenes, so on and so forth. Okay, that would demand then hierarchical patterns, because already right off the bat, Andrew, you have DNA differences, okay, within the genomes of living organisms and across, okay? So now you have DNA differences between humans and, let's say, the ape kind and the dog kind, okay, like your canids, your felids. So even if scientists were to come an hour after creation and start doing assessments or genomic analyses, they are going to see hierarchical patterns on a genomic level, okay? Even though this creation event was 1 hour prior. It demands hierarchical patterns. I mean, here's the last thing I'll say, and then I'll yield, and you can tell me what you disagree with. But even if you were to just look at these phylogenetic trees for the audience sake, okay? Let me see. Okay, so here's one thing I want to point out. Here's a diagram Rahmat and myself made, okay? You've got the tree of life, okay, that evolutionists, they assume all life is related through common ancestry based on these homologous patterns, nested hierarchical patterns in DNA and proteins, so on and so forth, okay? But then they assume that if the design hypothesis is true, that God would have created humans equally distant in terms of anatomy, morphology, physiology, and genetics with all other forms of life. I mean, that's very simplistic thinking, because by definition, humans would have to be, and tell me if you agree with this, humans would have to be more similar to some creatures, like the chimpanzees, the great apes, the old world monkeys, the new world monkeys, okay? And less similar to other forms of life, other creatures like fish, like whales, okay, which fall into the mammal category. So, I mean, the fact that we can actually categorize creatures and animals and biological life, the fact that we can actually do that is strong evidence for design, because we are looking at similarities in structure, similarities in DNA. So yeah, here's what I wanted to show, and then I'm going to yield, is right here. Even when you look at these hierarchy, these phylogenetic patterns in terms of hierarchies, okay, so you've got humans, okay, that are more similar to the chimpanzee, so they're closer on the tree, right? Because they share more in terms of similarity than they do with the mouse or than they do with the fish. I mean, even if you go down the line in these phylogenetic trees, the mouse is obviously going to share more with the chimpanzee than it is with the fish. And even if you go into the monkeys and the apes, yeah, the great apes and humans are going to share more with each other than they do with the New World monkeys, and they do with the lemurs. That's why we got to look to these differentiating lines of evidence. For example, the irreducible complexity that we see in these DNA elements that essentially should not really be there if common descent was true, like the pseudogenes, the taxonomically restricted genes known as orphan genes. I mean, you'd need 5 or 6 consecutive beneficial mutations just to evolve an orphan gene. I mean, orphan genes are highly essential to the specific organism. And it's never really been observed, this co-option. It's been assumed by looking at non-coding sequences versus coding sequences, looking at gene families, things like that, assumed, but never actually observed. So my question back to you is, why wouldn't we expect these hierarchies? Are you actually saying that God should have created humans equally distant from all other forms of life, or created apes or fish equally distant from all other forms of life? I mean, wouldn't naturally you have whatever creature you're looking at? Shouldn't naturally that creature be more similar to some and less similar to others? Isn't that just what are your thinking? What's your thinking on that?
Right. So that's a good point, and I think that gets into the idea that what best explains this nested hierarchy, and namely the fact that no, I don't think God would have created them all equally distant from each other. However, whether or not that's the case, if they were designed in one form or another and they did not share, if these created kinds did not share a further common ancestor down the line, I guess phrasing it poorly, but like you would basically see, I'm not showing my hands here, but like you would see one kind to the left, and you would see another kind, and then another kind, etc., etc. And then all of them, you would see kind of a polytomy connecting them in the middle. So that doesn't necessarily mean that there is a common ancestor, but it means that if they haven't shared this reproductive history, then we would expect there to be no unique nested hierarchy pattern, basically, for instance. So we would expect nested hierarchies, but we would not expect a statistically significant nested hierarchy, for instance. So,
well, what do you mean by statistical consistency when it comes to the hierarchical pattern?
meaning that if we were to take these organisms, and if common design were true, and we produced trillions upon trillions of trees, which is that those are all the possible trees that can be generated, even with a few dozen taxa. And you generate these trees according to common design, or at least we should expect from common design. This is the point that there would not be any correlation, significant correlation between any of the trees. So one would not dominate over the other, basically, or this particular group would not dominate over the others in terms of appearing.
What group is dominating another group over the others?
Well, like, okay, so imagine you have these possible trees, okay? So you have these taxa, and you're connecting them in different ways. You're assigning you're basically connecting them in different ways, and you're trying to find the one that fits the data the best. Okay, this is a standard phylogenetic method. Okay, now
real quick, Andrew, just hold your thought for about 30 seconds. I got to run upstairs for 1 second. This will be like a quick intermission, and then we'll continue right when I get back. Okay.
Okay.
All right, and we're back. Commercial break is over. I also just wanted to point out to the audience, because I was thrown off there because I just got slammed. I don't think I'm the only channel in the community that did, but I just got slammed with a bunch of copyright, not strikes, but claims. And I think because some of our videos have Genesis apologetics clips in it, and I think they're slamming everybody with copyright. I don't know if Raw Matt's in the chat, maybe check that out, brother, because it threw me off because I just got like a notification for like 20 emails. I'm like, what is this? Anyways, but we're good. I'm sorry to interrupt you there. So commercial break is over, and go ahead, my good man. We're still going on over an hour and 3 minutes. So we'll keep this going. If David Neff's in the chat, I'm going to send him a link as well. He can join whenever he's ready. We can talk about the things he wanted to talk about. And yeah, go ahead. Continue, Andrew.
Yeah, we can keep going for a few minutes, and then if David may or may not show up, but then we can open the chat for anyone who wants to join.
Right. So I think you're answering the question, what specific groups would dominate other groups over.
Right. So basically you're constructing all these possible genealogical connections between these taxa. So you don't know which one is the right one, basically. And when you do the analysis, the program you're using is going to spit out the consensus trees among them, like the trees that best match the data. And when you do that, you find that out of these trillions of possible trees, or at least many different possible trees, you get at least a few dozen that are consistent, or that are the consensus trees, basically. So that's what you get with common descent. For instance, you take a bunch of these different taxa and you construct all possible trees for them. And generally, this is what I mean by statistically significant, that out of all these possible trees, you're getting one that is consistent and matches the data. Whereas with common design, you would not necessarily expect that. And that's kind of the point here, is that we have good reason to suspect that that would be the case for common descent, that this would be statistically significant for one or a few possible trees out of this larger set, because naturally there is only one unique genealogy, basically, because of course Earth has only existed once, according to universal common descent. And that's what I mean by statistically significant, and the fact that we don't necessarily have the same reason for common design, because, like I've said before, we don't have the same kind of reproduction. So we would not see the statistical significance. We don't have reason to see the statistical significance under the common design model. So go ahead.
Okay, so good points. So what I say is I've looked at these number of papers on statistical nested hierarchies, okay, where they will look at certain groups and they will make a prediction that there should not be, statistically speaking, a hierarchy, let's say in the great ape group. There should not be a hierarchy if special creation and independent origins is true, if that model is more superior than the universal model. But in all of these statistical hierarchy assessments and analyses and in these papers, they make the erroneous assumption that creationists, for one, don't also expect hierarchies, but most importantly, that stems from the creationist assumption and starting point of what? Design diversity, right? Created nuclear heterozygosity, which does demand hierarchies. So where they are concluding that the independent origins model is false based on these hierarchical patterns, okay? It's actually based on a straw man. They are arguing against, and they are doing these analyses against what's called evolutionary creation. Okay, no, it's the evolutionists that explain all DNA differences as a result of mutations, while it is the creationist that explains the majority of DNA differences as the result of creation, initial design, and front loading. Now, this is why, in terms of these statistical hierarchies, we as young Earth creationists, we've made predictions. For example, Dr. Herman Mays, he looks to these amino acid trees. He looks to these statistical hierarchies in terms of third position codon variation, okay? Because there's a lot of redundancy in the genome, but it turns out that these redundant elements, the third position, this third position codon variation that we see in coding for protein, okay? It turns out that these positions don't only just tolerate error, okay? They're not there just for redundant purposes, but they are there for information flow. They are multifunctional. They are there for assistance and control, okay, in the slowing and speeding up of processes in the cell. These are things that evolutionists never predicted. They think that this neutral variation, okay, like I was saying earlier, that's why it all comes down to function. That's why it all comes down to genetics. Is this all really the result of mutations? Because evolutionists assume all differences are the result of mutations. A lot of these differences, okay, that are being looked at in terms of these statistical hierarchies, we are predicting that they are not actually the result of mutations. They are the result of initial design. And you can even look at hierarchical patterns in these highly conserved mitochondrial DNA proteins, for example, the cytochrome C, which the evolutionists would say, okay, and I debated conspiracy cats, why would a designer create these highly conserved mitochondrial DNA proteins, but why would he make them different across all these different species, right? Why is the cytochrome C mitochondrial DNA protein just a little bit different than the one in the chimpanzees and a little more different than the one in the old world monkeys, forming these hierarchical patterns? Well, we actually have evidence for what's called protein moonlighting, multifunctionality in these nuclear DNA proteins. So we're saying, okay, these hierarchical patterns in these highly conserved proteins in the mitochondrial DNA, they are also multifunctional, just like the multifunctionality seen in these nuclear DNA proteins. Now, we have not yet tested to see if these highly conserved mitochondrial DNA proteins really are multifunctional and involved in information flow and other essential mechanisms. So that's a testable prediction that we've yet predicted. But if it's true, once again, we see that these are there for a purpose, for a design. It comes down to function. I mean, you've heard people like Dan, Erica, they'll say that the majority of the genome has to be junk, neutral junk, no more than 20%. because that would be the case if evolutionary theory and universal common descent was true, because then the genome should reflect that. It should be mostly consisting of evolutionary leftovers, genomic fossils, okay, ancient viral infections like the ERV. So that's why it's a direct head-to-head prediction. And the evolutionists aren't stepping up and making predictions of their own. But I will say this, and I'll yield. Since we're on the topic of statistical hierarchies, we have done, and I'm going to share screen, in terms of baraminology, okay? And it might take for a second, I'm just sharing screen. So in terms of baraminology, this is from Dr. Matthew McLean, who we've had on, and they've done baraminological analyses, and they have found discontinuities. Okay, they have found discontinuities and barriers, okay, in groups, even within fossils. Okay, so he's finding discontinuities in the synapsids. He's finding it in the Dinosauria, where you've got continuity within these groups, and then discontinuities outside the groups based on essentially statistical analyses of different traits in terms of anatomy, morphology. And you got to look at enough traits, okay? So there's some papers if you want to look at it. Now, when it comes to fossils, though, and this should not be the case if universal common ancestry is true, you should see a very nice linear progression. You shouldn't find these discontinuities, okay? And I've got a ton here on the all the different Dinosauria groups. You can look at the discontinuities in terms of the mammal-like reptiles as well, but also when it comes to the genetics. Now, the fossils can be a little deceiving because without the genetics, we know, Andrew, and you can definitely comment on this, we know that today, oftentimes there's more variation within the same species than across species, okay? So when you're looking at just a fossil, it's hard to declare relationship because is this due to homology or is this due to convergent evolution? That's a problem even for the baraminologists, okay? That's a problem for the evolutionists too. But when we look to the genetic data, I'm not sure if you're up to date on this current study. This is the last thing I'll say, but there's been a study, a barcoding study, okay, where they discovered that there are genetic barriers in these mitochondrial DNA sequences. It's called barcoding. They're looking at the CO1 gene, highly conserved, and across pretty well all of life, they are finding low genetic diversity, and they're saying that all species today originated at the same time, okay? Which is consistent with our model, of course, of the bottleneck at the flood. But literally, they are saying, one author said, he fought the data as hard as he can because they literally say in the paper that, or is it here? They literally say in the paper that there are genetic barriers within species. And that's exactly what we'd expect if independent origins was true versus universal common ancestry. So are you familiar with that paper? And what do you think about these analyses in terms of fossil groups like the Dinosauria, like the synapsids that the baraminologists are doing, and they're finding discontinuities that should not be there essentially if universal common ancestry is true. And we've got this genetic data that says, wow, over 90% of species today originated at the same time due to low genetic diversity in this CO1 gene. So we got genetic data and fossil record data that points to one conclusion, and that conclusion being independent origins and separate ancestry. So I know I talked a lot there. You had a lot of good points that you brought up. Statistical hierarchies is definitely a technical topic. So yeah, what are your thoughts, Andrew?
So I guess a question I might ask would be this barcoding gene, was it or was it a single gene, or was it analysis based on multiple genes?
Well, they're using the CO1 gene, which is highly conserved across all these different species because the function of it is so important that essentially natural selection will conserve it, right? There's very little mutations in it, because if there is a mutation in something like a CO1 gene, then selection would wipe that creature out. But they've also looked at, to answer your question more specifically, yeah, they've looked at genetic diversity levels in nuclear DNA as well, which showed us that humans have low genetic diversity. We talked about that in our last debate. This is specifically about the mitochondrial DNA, which is a lot smaller than the nuclear DNA. But yeah, so for example here, in other words, each organism began with only one form of the barcode gene, and all the diversity has developed within that period. And what does it say? Why would all organisms converge at the same time frame? It just does not add up for the evolutionary theory. They tentatively suggest that the last ice age might explain the pervasive pattern. So here's the thing. Yeah, they've got explanations for the pattern, hypotheses, I should say. But the data fits nicely with our model, where species would have all originated at the same time at the flood event. We've also got the nuclear DNA data that suggests low genetic diversity in human beings, which makes sense since we have a starting point from two people. But yeah, there's multiple different genetic sets being looking at. This specific one is the barcode gene, in the mitochondria. Are you familiar with that study, or like what are your thoughts on it? It's strong evidence for the independent origins model. I don't want to say too much more about it because there's a lot of data flying around there, but
yeah, I personally haven't looked into it much. What's the date on it, if I may ask?
Of the actual paper, I think it was 2000 and either 18 or 17. I have the paper in here, the exact paper in here somewhere. But I got 1,000 different slides here. So, I mean, the whole point of the matter is we can look to fossils. We can look like for example, for you. Okay, so we're doing these analyses in terms of baraminology when we're looking at these fossil groups. But, even the creationists will admit, okay, so here's some of the work coming out of that. Here's a paper that you can study, okay, where you're looking at these discontinuities. But here's the thing. Genetics is still the most important. Okay, I would say that genetics is still more important than these baraminological analyses, okay? Because fossils can oftentimes be deceiving. Would you agree with that, in the fact that it's hard to differentiate between true homology and convergent evolution?
Sometimes, yeah.
Right. So, and that's why the historical reconstructions, Andrew, can oftentimes be up to so many assumptions, interpretations, and if the bigger picture is not questioned, but the empirical data is contrary to the historical reconstructions, that's where those historical reconstructions really are just that. Reconstructions, stories. But well, here, so we are going close to an hour and 20 minutes, and you're probably getting tired too, since we'll be going at this nonstop. Why don't we, since you're the guest again, why don't you respond to anything that I brought up in terms of these discontinuities in this work being done in baraminology, as well as the genetic data? Anything you want to respond to, take the floor, and then we'll open it up. I don't see David Neff in here, which means maybe he fell asleep or something. I don't know. I think he does have to work. So then we could just open it up if other people want to chat. But yeah, go ahead, man. I appreciate you always being willing to have these cordial discussions, cordial dialogues, and go ahead. What are your thoughts? And take your time.
Right. So I guess just to summarize my position, I think that compatibility by itself is not a reason why nested hierarchies are evidence for common descent. Rather, we have the mechanisms of how common descent would produce such patterns, because we see these in living organisms today, like living populations, for instance. We do not have the same thing for creation, which kind of gets into okay, what's the best explanation? What's the supernatural versus natural explanations, etc., etc. But I think the main point I would say is that compatibility by itself is not evidence. We need the consistency. We need independent lines of evidence. We need these specific predictions. We need mechanisms that could explain these predictions. And although some of it is consistent with creation, I don't think all of it, or at least as much as common descent, is fulfilled by creation. So I guess I'll just leave it at that for now, and we'll see who pops in.
Awesome. Well, I appreciate that. It's been a good discussion, and these are some of the more heavy topics in the whole ancestry controversy, right? The nested hierarchies and getting into the nitty gritty of the genetics. Because I always point out we understand so little of the DNA language. So oftentimes the assumption that the evolutionists make and have made throughout history, the history of this theory. I mean, first it was the vestigial organs, then it was the junk DNA paradigm, and oftentimes they're found with egg on their face. And I'm not personally seeing any real future testable predictions coming from the evolutionist side. But in terms of the creationist side, we're looking at the genetic related data. We're looking at the nested hierarchies, and we're wondering, is this a reflection of descent or is this a reflection of design? Therefore, we're saying those hierarchies would be there for purpose, for design, for function. And so on a genetic level, that's what we would expect to find. And you can find prediction after prediction coming from young Earth creation PhD scientists on very, very specific predictions in terms of nested hierarchical patterns in DNA. Because the DNA, obviously, the phenotype's a reflection of the genotype, a reflection of the information system. So that would mean we should go down on a molecular level, and you see the consistency of hierarchical patterns. So that's where we would best make those predictions. But yeah, I don't think David is going to be joining. So if you want, now I know you said that we're going on about an hour and a half. Do you want to open up the mic, or did you want to call this one quits, have it its own stream, and then maybe open up a separate one for open mic? Oftentimes open mics can turn out to be 4 or 5 hours. So maybe since this is an hour and a half, maybe we'll let this one stand as the way it is, and just I'll open up another stream for an open mic. Does that work for you, empiricist? And no obligations. You don't have to join. I know we just went at it for an hour and a half. So yeah, I think yeah, go ahead.
Oh, I was just gonna say I can probably stay for like half an hour. So if you wanna open up the mic, I guess now, and then I can drop out after a little bit, and then you guys can have at it if that works for you.
All right.
Because I'd really like to see, in particular, like Roof and the Dot is in the chat here, and we've kind of discussed some things in the live chat, but I'd really like to hear your thoughts on this. But no obligations for anyone in the chat to come in. Just I want to hear some opinions.
What I'll do is I'm going to shut this stream down, leave it as it is. I'm going to right away open up an open mic, send the link into the live chat. We'll see how many people we get into the back room, and then we'll go live again. Just for the odd chance that this goes for 4 hours, our initial hour and a half conversation might get diluted down and all that. So I'm going to shut this down right away, guys. I'm going to open up another stream. It's going to be an open mic. Whoever wants to join, I'm going to throw the link into the side chat, and I'll give David Neff 20 more seconds, and then I'm going to shut it down and open up another. So James Downard says, actually, RJ is moving stuff at his house and is only occasionally. RJ, you're always welcome to join in, make your case, bring up your points, your arguments. I like these cordial, respectful discussions. So we can even talk about your synapsids. I'd like to hear James Downard's opinion on the discontinuities that we're finding in terms of baraminology. So Rue from the Dot says he's going to go back and rewatch the first 1.5 hours. Yeah, 90 minutes has flown by, Andrew. It's been a great talk. I'm going to shut it down right now, and we're going to open it up for open mic. If you want to join for 30 minutes or however long you want, you're always welcome on the channel and in the shows. So everybody, God bless, and I hope you enjoyed the discussion. We're going to have many more of these. Saturday, me and David Beldock will be doing our debate. Same time. Let's hope and pray he feels better. And SFT's out.
Stay curious.